All right, let's begin to talk about the most important concept of modern biology. This idea is known as the Central Dogma. The idea is that DNA holds the information of how to rebuild itself and how to build proteins. RNA uses that information to build proteins, and those proteins carry out the functions of the cell and the organism. Originally, this was imagined to be a one-way street, that information flowed only from DNA to RNA to proteins, and for the most part, that is the case. However, modern biology has found some notable exceptions to this directionality, but those are beyond the scope of this course. So, the first part of this concept is that DNA directs its own replication. We discussed this in the last section. DNA helicase opens up and unwinds the DNA double-stranded molecule, exposing the two sides. Then DNA polymerase comes in and begins to add complementary bases to cover the exposed strands, so that where there is an A T will be added, when there's a G, a C will be added. This works very quickly on the leading strand, which is in the correct orientation, but in the incorrectly oriented lagging strand, short sections of Okazaki fragments are laid down. Then DNA ligase joins together any sugar phosphate bonds that haven't yet formed. Now let's meet the other nucleic acid in the central dogma, RNA. This nucleic acid uses a different sugar to make it up. It uses ribose instead of deoxyribose. This makes it behave quite differently than DNA. It exists as a single strand instead of a double helix, although it does have some secondary structure and uses similar base pairing strategies. RNA also uses a slightly different base than T. It is known as U. The other three bases are the same names as in DNA. RNA comes in three different forms, each an important part of building a protein. The messenger RNA is used to take the instructions indicating the order of amino acids and proteins to the cytoplasm. The transfer RNA is able to translate the nucleotide code into amino acids to be joined together. And the ribosomal RNA holds the structure together while it's being built. The final of the polymers referenced in the central dogma are proteins. Proteins are polymers made up of different combinations of the 20 common amino acids. These molecules do most of the work we've discussed all year long. They are the enzymes and structural molecules that build an organism and make it run. So the key to the central dogma is that DNA directs its own copying in the process of replication. The information is changed from DNA to RNA in the process of transcription, and the messenger RNA created along with transfer RNA and ribosomal RNA are used to create protein polymers from that information in the process of translation. Again, this is generally a unidirectional process. RNA doesn't usually tell DNA how to copy or change its order. Proteins don't direct changes in RNA or DNA sequences or tell how to build themselves. But like all dogmas, there are exceptions, some of which become very important in human health and disease.